Imagine a burglar, right? They break into this highly secured bank,
Mm-hmm.
and usually the very first thing they do is look for the alarm wires and just physically cut them to disable the system.
Right. The classic movie heist trope.
Exactly. But imagine this burglar does something completely different. Instead of cutting wires, they deploy this tiny, like, invisible sponge.
A sponge.
Yeah, a sponge. And it's so perfectly engineered that it absorbs the exact frequency of the alarm siren. So the alarm is screaming, the lights are flashing, the system is technically working.
But the sound is just trapped.
Exactly.
Yeah.
The police outside hear absolutely nothing.
That is, I mean, it's the perfect microscopic heist. The defense mechanism is firing on all cylinders, but the signal just never reaches the police, or in our case, the effector. It's completely neutralized by physical sequestration.
And that is exactly what is happening right now at a molecular level inside the bacterial biome. Welcome to today's deep dive, where we're unpacking a wild new 2026 paper out of the journal Science by Tal and colleagues.
It's a fascinating paper.
It really is. We are going to figure out exactly how viruses pull off this exact heist. It's this invisible arms race between bacteria and the viruses that infect them, which are called phages.
Right.
And our mission today is to look at how scientists used advanced AI, specifically structural modeling, to find these secret viral weapons that just completely disarm bacterial immune systems.
Yeah, and to really appreciate the viral countermeasure, the burglar sponge, we first have to understand the sheer sophistication of the bacterial alarm system itself.
Right, the bank's security system.
Exactly.
Yeah.
Because bacteria are not just, passive bags of protoplasm waiting to be hijacked. They have incredibly intricate innate immune pathways. Two of the most fascinating ones highlighted in this research are the Thoeris and CBASS systems.
Right, and I found the mechanics of those pathways just incredible. It's essentially this biological domino effect. So the phage lands on the bacterium and injects its viral DNA, right?
Yep.
But the bacterium doesn't just sit there. It detects the invasion using a sensor protein.
Right, like a security camera.
Yeah, exactly. And in the Thoeris system, we're talking about a sensor with a TIR domain, which basically acts like an emergency manufacturing plant. It immediately starts pumping out these highly specific chemical signaling molecules.
The alarms.
Yes. For type I Thoeris, that's a nucleotide derivative called 3C-ADPR.
Right.
And for CBASS, it produces a different one, 3-FIT3C GAMP.
Yeah, and those nucleotide molecules are literally the sirens from your bank heist analogy. They act as intracellular second messenger.
Second messengers, right?
And because they are so small and highly diffusible, they just rapidly flood the entire internal volume of the bacterial cell.
Oh, wow.
Their sole purpose is to travel through that space and physically bind to an effector protein-like TSA in the Thoeris system.
Okay, so they find the effector.
And once that effector binds the nucleotide signal, it undergoes this massive conformational change. It wakes up, basically,
Yeah.
and triggers a catastrophic defensive response.
And that response is what really caught my attention, because the effector triggers regulated cell death. It completely halts cellular growth and just kills the infected bacterium.
It does.
Now, playing devil's advocate here for a second, from an evolutionary standpoint, suicide seems like a pretty catastrophic failure of an immune system, doesn't it? I mean, why not just deploy an enzyme to chop up the invading viral DNA and survive?
Well, it really comes down to a matter of speed and numbers. Clarifying efficiency.
Right.
By the time an enzyme tracks down and degrades every single piece of viral DNA, the phage might have already assembled like 100 new copies of itself.
Oh, so it's just too fast.
Way too fast.
Yeah.
So you have to shift your perspective from the individual bacterium to the whole colony. Bacteria live in these densely packed communities of billions.
Okay.
This regulated cell death is actually an evolutionary strategy of altruistic suicide.
Altruistic suicide.
Right. By rapidly destroying its own cellular machinery, the infected bacterium denies the virus the factory it needs to replicate.
Ah, I see.
The cell dies, the virus is trapped and degrades, and the millions of neighboring bacteria survive.
So it's a microscopic scorched earth policy. The brutal math of colony survival. Let's use a fire alarm analogy for you listening at home.
Oh, I like that.
So the phage is the fire. The sensor is the pull station on the wall. The nucleotide signals, the 3C-DPR and CGAMP, are the loud, ringing noise of the alarm.
Mm-hmm.
And the effector is the sprinkler system that just completely floods the room, ruins everything, but it stops the fire from spreading to the rest of the building.
That is a perfect way to visualize it.
So just do a quick recap for you listening to lock this in before we move on.
Good idea.
Bacteria use these chemical alarms, right, these nucleotides, to trigger cell death and stop viral spread. That's the core mechanism.
Exactly.
And I want to throw a question out to you, the listener. What stands out to you about this strategy? It's this fascinating, brutal math, right? Sacrificing the one to save the billions.
It's very dramatic when you think about it.
It really is. But it's an airtight defense, assuming the signal actually reaches the effector.
Right. Which brings us to the viral counterattack. Because viruses are the ultimate evolutionary puzzle solvers.
Right.
They haven't just, accepted this altruistic suicide strategy.
I've seen that.
Right. They've developed a class of anti-defense proteins, specifically a subset that scientists call the sponges.
The invisible, sound-absorbing sponges from our bank heist.
Exactly. And I know researchers had previously stumbled across a few of these, like there are families named TAD1, TAD2, and AZB2.
But the literature notes those were found almost entirely by serendipity, right? Like just by accident.
They were, yeah. Complete luck. And what those sponge proteins do is they physically bind to the bacterial alarm molecules, the 3-CEDPR or the integramp, and they just sequester them.
So going back to our analogy, the virus essentially wraps a soundproof box around the ringing fire alarm bell.
Yep.
The sensor is still producing the nucleotide alarms, but the viral sponge traps them before they can diffuse across the cell and bind to the effector.
Right, so the sprinklers never turn on.
The house doesn't burn down, and the virus gets to happily hijack the cell, replicate, and eventually burst out to infect the whole colony.
It's brilliant.
Yeah.
But the challenge for the scientific community was that finding more of these sponges using traditional methods was, well, virtually impossible.
Why is that?
Usually, if you want to find a specific type of protein across different genomes, you look for homologous genetic sequences. Like you look for similar spelling in the DNA.
Okay, makes sense.
But viruses mutate at staggering rates to escape bacterial defenses. If you line up the genetic sequences of the known sponges, they are vastly divergent. They share almost zero obvious sequence similarity.
So the text search fails. And this is where the researchers made this brilliant pivot. Here's where it gets really interesting.
Yeah.
They hypothesized that even though the underlying genetic code was totally scrambled by mutation, the final 3D physical structure of these proteins had to remain essentially identical.
Form follows function.
Exactly. If the goal is to trap a very specific nucleotide molecule, you need a very specific physical trap.
It's the principle of convergent evolution operating at the nanometer scale.
Mm-hmm.
So the researchers looked at the few known sponges and established this distinct structural fingerprint. And they noticed 3 non-negotiable architectural traits.
Okay, what were they?
First, the proteins themselves are tiny, typically under 100 amino acids. Second, they form homo-oligomers.
Meaning they utilize this really highly efficient modular architecture like Lego pieces.
Right, like Lego pieces.
They take that one tiny protein sequence and assemble multiple identical copies of it into a larger functional complex.
Exactly. They form dimers made of 2 identical units, tetramers of 4 or hexamers of 6. And this is an incredibly elegant evolutionary solution to the limited carrying capacity of a viral capsid.
Right, because viruses don't have a lot of room.
A virus has precious little genomic real estate. So instead of encoding one massive complex protein, it encodes a single tiny subunit and just lets thermodynamics drive the assembly of a much larger symmetrical cage.
That is so smart, which
Yeah.
perfectly sets up their third structural fingerprint, the binding pocket.
Yes.
When these identical Lego subunits snap together, the interfaces where they meet form these deep internal void spaces. And crucially, those pockets are lined with positively charged amino acids.
Which is key.
Right, because the bacterial alarm molecules, the nucleotides, have that dense, negatively charged phosphate backbone.
Opposites attract.
Exactly. The electrostatic attraction literally pulls the alarm directly into the void space.
Those pockets have a very specific internal surface area, usually mapped between 200 and 700 square angstroms, perfectly tailored to swallow those specific nucleotide signals.
Okay, so let's do another quick recap here.
Okay.
We've got our second bullet point for you. Viruses use these structurally similar sponge proteins with positively charged pockets to trap negatively charged immune signals.
That's the summary, yeah.
And here's a question to chew on. If you knew the exact physical shape of a lock, could you find every single key that fits it, even if the keys were made of totally different materials? Because that's exactly what these scientists set out to do next.
It is. because now they have a highly specific physical blueprint. They are looking for tiny, modular, positively charged cages.
But how do you find that physical shape hidden inside millions of uncharacterized genetic sequences? Like we said, you can't just run a text search.
No, you need a massive computational pipeline, one capable of bridging the gap between a raw sequence and a 3D physical reality.
And this is where AI comes in.
Big time. The team utilized AlphaFold Multiprimer, which is an advanced iteration of the AlphaFold AI.
Right.
They took a staggering data set, roughly 32 million non-redundant, completely unannotated proteins.
32 million.
Extracted from 2 million different phage genomes.
The scale of that is just mind-bending. 32 million mystery proteins. And AlphaFold isn't just looking at the linear sequence, right?
It uses coevolutionary data. It analyzes which amino acids mutate together across evolutionary time. That allows it to accurately predict how these peptides will fold in three-dimensional space. And more importantly, how they will assemble into those multi-subunit homo-oligomers we talked about. But once AlphaFold predicted the multi-subunit structures, the researchers didn't just stop there.
What did they do?
They ran those 3D models through specialized pocket prediction algorithms, programs like CASP and AutoSite.
Okay, so CASP and AutoSite.
Right. And these programs scan the topography of the assembled protein. They map the surface area and the electrostatic charge of those internal voids. They were literally hunting for the physical dimensions of a molecular bear trap.
That is so cool. But I read they also leveraged a really clever evolutionary shortcut alongside the AI, right? Something to do with gene fusions.
Oh, yeah. This was brilliant. They noted that some of the known sponges were occasionally encoded as a single fused continuous protein in certain viral genomes.
Wait, why would viruses fuse proteins together?
It's an evolutionary Swiss Army knife.
Okay.
See, bacterial colonies are highly heterogeneous. A single bacterium might deploy multiple different alarm systems, say Thoeris producing 3C80PR and CBASS producing CGAM.
So the virus needs to block both.
Exactly. By fusing two structurally distinct sponges into a single gene product, the virus ensures that both countermeasures are deployed simultaneously and in equal measure.
Oh, I see. And assuming guilt by association, the researchers started looking for uncharacterized proteins that were physically fused to known sponges.
Right.
And that led them to a massive discovery. They found this ubiquitous family of short proteins, historically just labeled as PF21825, constantly fused to known defense evaders.
And they renamed this family Sequestin.
Sequestin. Catchy.
Very. And when they ran Sequestin through the AlphaFold pipeline, the structural prediction was stunning. AlphaFold confidently modeled Sequestin forming dimers or tetramers, creating a pocket that perfectly accommodates the Thoeris alarm molecule, 3Z80PR.
But, a computational model, no matter how advanced it is still just a hypothesis until you validate it in the actual lab.
Oh, absolutely.
And their validation was incredibly thorough. They synthesized Sequestin, purified it, and physically incubated it with the Thoeris alarm molecules.
Yeah, they used high-performance liquid chromatography, or HPLC, coupled with size exclusion chromatography.
Let's break that down a bit.
Sure. So this technique allows them to physically separate molecules based on their size and weight.
Okay.
And they demonstrated that the small, free-floating nucleotide alarms completely disappeared from the solution.
Just
Vanished. Instead, they co-eluted perfectly with the massive Sequestin protein complex.
The sponge soaked them up.
It did. And to prove it wasn't just destroying the molecules, they used chemicals to denature, or basically unfold, the Sequestin sponge. And guess what? The intact alarm molecules spilled right back out.
It was definitively holding them hostage.
Definitively.
But here is the detail that absolutely floored me. They didn't just find Sequestin in obscure, newly discovered phages.
No, they didn't.
They found it in the T4 phage.
Yeah, which is huge. T4 is arguably one of the most famous model organisms in the history of molecular biology. I mean, scientists have been tearing apart its genome and studying its mechanisms for the better part of a century. It was instrumental in proving that DNA was the genetic material in the first place.
And yet, for decades, T4 had this protein called Y16Q that was just sitting in the database labeled unknown function.
Just sitting there. And thanks to this structural pipeline, we now know Y16Q is a Sequestin sponge. It's a highly specific inhibitor of the Thoeris defense system.
Wow.
When the researchers engineered the T4Y16Q protein into a completely different virus, that new virus suddenly gained complete immunity to the Thoeris defense.
It was hiding in plain sight the whole time.
Yep.
It really highlights how much of the microscopic world remains totally dark to us because of our reliance on just sequence-based text searches.
The paradigm shift.
Okay, so let's hit our third quick recap.
Let's do it.
Using AlphaFold, scientists found Sequestin in a sponge hiding in plain sight, even in historically famous viruses like T4.
That's the big takeaway there.
And here is my question for you listening. Think about that for a second. A virus studied for generations had a secret weapon staring us right in the face. What other secrets are hiding in the things we think we already understand completely?
That's a great question. And, Sequestin wasn't the only structural architecture they uncovered.
Wait, there's more.
Not by a long shot. The AlphaFold pipeline revealed architectures that were significantly more complex. The most striking example is a massive family of proteins they designated as Lockin.
Lockin. I spent a lot of time looking at the structural models for Lockin in the paper, and it is just a masterpiece of molecular engineering.
Isn't it?
It doesn't look like a simple clamshell or a basic pocket. It looks like a highly engineered mechanical cog.
Yeah, Lockin forms a hexamer. 6 identical protoners locked together to create this rigid, symmetrical ring.
A 6-piece ring.
Exactly. And its potency is remarkable because it inhibits both type I and type II Thoeris systems.
Why, both of them?
Both. It's capable of sequestering the type I signal, 3C-ADPR, as well as the type II signal, which is a structurally distinct molecule called his ADPR.
So it can trap up to 6 alarm molecules simultaneously, slotting one into each of the deep pockets formed between the teeth of the cog.
Right.
The researchers actually achieved a crystal structure of Lockin mapped down to a 1.6 angstrom resolution.
Which is incredible.
That is a level of clarity where you are essentially watching individual atoms interact.
At that resolution, you can see the precise molecular mechanics of the evasion. Inside the Lockin void, highly specific amino acid side chains reach out, almost like fingers to grasp the alarm molecule.
Little fingers.
Yeah. You have amino acids T47 and Q59 that actively read the chemical structure of the adenine base. This ensures it only traps the correct target.
So it's not just some generic sticky void. It is a custom-molded cradle.
Exactly. And then you have amino acids K45 and K48, which are positively charged. They reach out to stabilize that negatively charged phosphate backbone of the alarm molecule.
Oh, wow.
It's a multi-point vice-like grip.
The precision is absolute. And the researchers proved this dependency by selectively mutating those specific amino acids, right?
They did, by altering K35 or K48, effectively changing the shape of just one or two of those fingers inside the trap Lockin, completely lost its ability to sequester the signal.
Just broke the whole thing.
Completely. The bacterial alarm reached the effector, the cell triggered its altruistic suicide, and the virus was destroyed. The trap was broken by altering a single atomic interaction.
That level of experimental validation is just incredible to me. But this pipeline got me thinking. We've been focusing entirely on sequestration, right? Sponges hiding the alarm. But evolutionary pressure rarely bets on just one strategy. In a dynamic arms race, sequestration can't be the only trick in the viral playbook.
You're exactly right.
Because I read that when they looked at phages infecting Streptomyces bacteria, they found a protein family called ACB5 that operates on a totally different wavelength.
Yes. ACB5 is a perfect example of the pipeline's versatility. ACB5 was identified because it shares that fused gene architecture and features a functional pocket.
Okay.
But it is unequivocally not a sponge. It is an active enzyme.
Oh, so instead of hiding the signal, it actually destroys it.
Precisely. ACB5 specifically targets the CBASS immune system. When the bacterial sensor floods the cell with the 3'3" cGAMP alarm molecules, the ACB5 enzyme binds to them and actively cleaves the phosphodiester bonds.
It chops them up.
It enzymatically degrades the alarm into inert, harmless fragments.
So returning to our bank heist analogy, if Lockin is the soundproof box placed over the siren, ACB5 is the burglar walking up with a pair of heavy-duty wire cutters and physically snipping the alarm system to pieces.
That is the exact difference, yeah.
Both strategies, sequestration, and enzymatic degradation achieve the exact same goal. They prevent the signal from reaching the effector, but they represent entirely distinct evolutionary solutions to the exact same problem.
And what's truly revolutionary is that both of these vastly different tools were discovered using the exact same structure-guided computational pipeline.
Because it's shape-based.
Right. By searching for functional shapes rather than genetic sequences, the methodology is totally agnostic to the mechanism. It simply finds the proteins interacting with the immune pathways, whatever they might be doing.
Okay, time for our final mini recap here.
Yes.
Lockin uses precise amino acid fingers to trap alarms, while ACB5 acts as chemical scissors to destroy them completely.
Beautifully summarized.
And a quick question for you. If you were designing a counterdefense, which strategy would you bet your life on? Hiding the evidence or destroying it completely?
Oh, that's a tough one. Destroying it feels safer, but nature loves redundancy.
It really does. Which naturally leads us to the overarching question for this whole deep dive. Why does a microscopic turf war between a bacterial colony and a phage actually matter outside of a petri dish?
Right. Why should we care?
Yeah, why should you or I care about CBASS and Thoeris?
If we connect this to the bigger picture, we should care, because the echoes of this ancient war are operating inside our own bodies right now.
Wait, really? In human cells?
Yes.
Yeah.
The human innate immune system relies heavily on a pathway known as CBASS sting.
CBASS sting.
Right. This is the primary mechanism our cells use to detect cytosolic DNA from invading human viruses like herpes simplex, or even DNA leaked from tumors.
Oh, wow.
And once it detects it, it triggers a massive inflammatory response to fight it off.
And the connection is that CBASS sting didn't just, spontaneously appear in mammals.
Not at all. It directly evolved from these ancient bacterial CBS systems.
No way!
Yes. The underlying architecture, a sensor detecting an invasion, producing a nucleotide second messenger like CGM, which then diffuses to activate an effector. That is a highly conserved evolutionary chain spanning billions of years.
That is wild.
The rules of engagement were written in bacteria.
So by stripping away the genetic noise and understanding the structural physics of how phages disarm these ancient pathways, we are fundamentally learning how our own immune system can be compromised.
Exactly. The implications for biomedicine are massive here.
I bet.
The structure-guided AI pipeline developed in this science paper is completely independent of prior genetic annotations, which means it can now be aimed at vast databases of eukaryotic viruses.
Like viruses that infect plants, animals, and humans?
Precisely. We can actively hunt for analogous immune-evading proteins that are modulating our CGS sting pathway.
So if we can map the molecular traps that human viruses are using, we could potentially design drugs to jam those traps, right?
Exactly. Forcing the virus to remain visible to our immune system. It opens up entirely new vectors for antiviral therapeutics and, honestly, even cancer immunotherapies.
That is unbelievable.
This shifts our posture from reactive observation to proactive discovery. We are no longer just waiting to stumble across these mechanisms by accident. We have the computational framework to map the dark matter of the viral world.
Which leaves us with a fascinating and honestly slightly unsettling thought to chew on as we wrap up.
Uh-oh, let's hear it.
We've just spent this time exploring how phages use highly specific, AI-predictable, microscopic sponges and wire cutters to silence bacterial cries for help.
Yeah.
Knowing that human cells share this exact same ancient alarm architecture, it really makes you wonder, what invisible, structurally uncharacterized sponges might human viruses be deploying right now inside our own cells, quietly absorbing the alarms of chronic infections while we remain completely unaware?
That is a chilling thought. But the tools to finally illuminate those hidden mechanisms exist today. Now, the scientific community just has to run the search.
The microscopic arms race is a lot louder and vastly more complex than we ever imagined. Thank you for joining us on this deep dive into the hidden world of structural biology. Keep exploring, keep asking the hard questions, and we will catch you on the next one.